Okay, this is the final prep for lecture 24. And you can maybe let me know. I've been doing generally one kind of longer recording, but if several smaller ones is a better idea for you, just let me know because it's easy enough for me to just do them this way compared to doing one really long one. And this is where we ended up with the distal visceral endoderm and anterior visceral endoderm being responsible. There's migration towards the anterior, and this endoderm is actually the source of signals that inhibit Wnt and nodal signaling, specify the anterior ectoderm, which is going to form forebrain, for example, and then the initiation as well of migration of cells around to form the primitive streak. Primitive streak formation is inhibited by the DVE and AVE, which means the streak is kind of forming at a default position and marking the posterior. So despite the fact that when you're observing the embryo, the first sign you see of anterior versus posterior specification is when the primitive streak starts to be noticeable, 6 to 6.5 days in post-fertilization. There are signals that have taken place in advance of this that make this the spot where that gastrulation is going to start. So anterior visceral endoderm, this is the area of migration. These are the inhibitory signals against Wnt and nodal. And then this is the migration of cells that's going to be moving around to form the primitive streak, which is a thickening of cells. These cells, the primitive streak is forming as the cells kind of continue to migrate around, and they begin to, if you remember, I don't see how I can possibly draw it here, but they start moving in between the ectoderm that's on the inside and the endoderm that's on the outside. So they migrate around in between that. But there is some extra embryonic mesoderm also that moves up into the sort of the region where implantation and placenta formation is going to happen. And this organizer, the node, is forming sort of just before the distalmost position at the cup. Okay. Now there are some genes being expressed here, but I think that we'll just leave it at this. Brachyury, which you've heard about, nodal, which you've heard about, Wnt and goosecoid, which you've all heard about, are doing similar jobs in this stage as they are in other organisms. So yeah, so I think that's about, and then the other thing is just that, like, this is the beginning of forebrain development, and it gets, with each day or half day that the process proceeds, we see further development. So there's going to be more gene expression and movement and rearrangement of cells to form, the anterior ectodermal structures and so on. Okay. So, and this is a useful table, and it just shows you that in Xenopus and mouse, the way the gastrulation is proceeding looks quite different. But the genes that are involved in these processes, brachyury and goosecoid, remember brachyury is for the whole of the mesoderm, and goosecoid is specific to the organizer region. That is the same for the mouse gastrula. And the secreted proteins, nodal related, and nodal cordon and noggin, Cerberus, and Cerberus related. So, and they're doing the same kinds of things. So nodal TGF beta family member, noggin and cordon antagonize nodal and other genes. Cerberus antagonizes nodal and some other Wnt and some other molecules as well. And so if you just remember what these guys do, and just remember what they're doing in the frog, they're doing practically the same things in the mouse. Okay, and so when we take a look at the fate maps, the overall fate maps of the chick and the mouse, now the mouse is flattened out because it's hard to view the cup fate map the same way. But this is the cup, and when we look at the cup, the anterior part is usually here, and the posterior part is usually here, and we're generally looking at it along the side. Whereas with the chick embryo, we're usually looking from the dorsal side down. So this is, we've seen this a lot. This is very unusual way for us to look at it, but you can see kind of the relative similarity. You've got your ectoderm, your mesoderm. The endoderm isn't very visible in this view because it's below. It's down below where, continuous with the yolk sac at least at the beginning of that. And then some of the extra embryonic mesoderm, which is shown here. These are ones that are going to, again, contribute to placenta. They're going to participate in the formation of other structures that are needed for support, for gas exchange, for nutrition, and all that sort of thing. And the node, where the notochord forms, right? They're the same. This is Henson's node. This is just the node. They're not in exactly the same place because remember that in the mouse, cells move anteriorly through the node, and the node kind of stays in place, whereas the node forms in the chick towards the anterior, but then it moves back towards the posterior as development proceeds. So, they're very similar structures, but the actual movement of cells and so on is a little bit different. One of the things we really want to emphasize, though, is the similarity between different organisms, because a mouse and a chick don't look particularly similar to each other, and yet, at these early embryonic stages, the embryos have many, many striking similarities. So if we look at the primitive streak at the most anterior part, the farthest that it reaches, the notochord forms, and then there is this region, the precordial head mesoderm. This is the mesoderm that's going to participate in formation of the head structure above the notochord, because the notochord doesn't extend right up into the head. It starts kind of approximately where the brain and the rest of the neural tube will form with each other. And then there are somites, and the somites give rise to, skeletal muscle, dermis, and the vertebrae, right? And then a little bit further down towards the posterior, we're seeing the more ventral mesodermal fates, like kidney, heart, blood vessels, and so on, certain kinds of connective tissue and that sort of thing. So from anterior to posterior, from the anterior, we're also seeing mostly dorsal mesodermal structures, and towards the posterior, we're seeing the development of ventral mesodermal structures. And so here, they don't, so if you were thinking of, like if we were looking straight at the head, the dorsal mesoderm stays up close to the dorsal side. These ventral mesoderm, the extra lines here are just letting you know that it moves further down into the body, because if we were looking at the body, suppose this was the anterior end, left, right, dorsal, ventral, in the belly region is where, the more ventral structures are going to form. Okay, so they're going to migrate to these new positions as elongation occurs. So it's kind of unusual, but if you think about the frog gastrulation, there's a similarity to this as well, because remember that the dorsal and the posterior pole is practically the same as gastrulation begins, but then as gastrulation proceeds and the cells are moving around, then you have your anterior, posterior, and your dorsal ventral, at right angles to each other. So as the body starts developing, the cells will rearrange themselves and move to their final positions. And so, and I've talked about this before, but the regression of Henson's node is coincident with the formation of the head process, which is going to form the head, and then the beginning of the formation of somites. We're going to spend a lot of time on the 90-minute repeating cycle of gene expression that kind of moves down through the body axis as the node regresses. And there's a population of stem cells that's just ahead of the node. And as the node moves through them, the stem cells move, along. And we'll be looking at that in more detail too next time. And just a reminder that the order of formation of structures as the node recedes is the notochord first, and then the neural plate and folds, then the neural tube as the tube starts to close, and then somites. And we've talked about that in a previous lecture. Now it turns out, this is quite interesting, again, another indication of how very similar the organisms are. So you take the animal cap from a frog embryo, and you excise bits of the epiblast of the chick embryo. So this is the part that's above the subgerminal space. You can take tissue from different parts of an embryo that has got the primitive streak form, and Henson's node has formed but hasn't begun regression. And you can do this experiment where you take that epiblast cells and you sandwich them kind of in between two chunks of Xenopus animal cap tissue, and then you observe what kinds of gene expression can you document. And the question is, can you induce the formation of neural tissue in a different species by culturing these tissues? And it turns out if you use either non-streak epiblast or streak epiblast that's far away from where Henson's node is, and you culture them, and you look for the expression of specific neuronal genes, you can't detect them. You can detect the control expression of EF1A, elongation factor. It's one of those kinds of genes that's expressed in all cells. So this is just to make sure that your methods are working. You have to show that you can detect gene expression where it's occurring, that it's not just some weird artifact of your work. But then if you take Henson's node and excise it and culture it between two bits of animal cap cells, you can actually get the expression of neurotropic factor 3 and NCAM, which is I think a neuronal cell adhesion molecule, if I remember that correctly. And these are specific to nerve tissue. And if you looked at the frog later on at stage 30 and you were to culture neural ectoderm cells and assess the gene expression, you would find those same genes being expressed in presumptive neural tissue of the actual Xenopus, the frog embryo. So this tells us a couple of things. One is that this is definitely an organizing center and inducer of gene expression, and that the signals are so similar between the two organisms that you can take the Henson's node of the chick, culture it with some frog tissue, and get the induction of neural fates, specification of neural tissue. It's pretty interesting. And then both in the mouse and the chick, but it's studied a lot more often in the chick because the chick embryo is just more accessible. But there is this zone of prospective stem cells, and it expresses this gene SAX1. That's one of the marker for these structures. And as the Henson's node is regressing, and just before the somites start to form, you can see this zone expressing. And then as it's moving along, the cells behind it are called neurogenic. These are the cells that are going to form the spinal cord. SAX1 is another one of the kinds of genes that is expressed specifically in presumptive neural tissue, right? So the somites that are forming are a little bit below the surface because they are blocks of mesodermal tissue, but these stem cells, as the node is regressing and the neural tube is forming, just ahead of where the node is, you have these neuronal stem cells, and they are preparing the tissue to form neural fates. And I think that might be everything. We're going to spend, as I said, a whole bunch of time in the next lecture talking specifically about sort of somite induction as well as some of the other signals and so on. So that might be as much as we need at this point. So next time we're going to go into this whole process in more detail, including some manipulation experiments that show that this material here, the pre-somatic mesoderm, quite a long time before it actually forms the somites, it already has been sort of specified anterior versus posterior, and that's pretty interesting, the whole process. And then neural crest cells. These are really interesting. Some researchers consider them a separate germ layer, separate from ectoderm and mesoderm, because they are formed from both types of tissue, and they do some very unique things. And we'll spend some time on that next time.